everyone. Thank you all for joining us for our June Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Hereditary Disease Foundation. I want to start by thanking our sponsor for this webinar, Neurocrine Biosciences, and also thank you to everyone who supports the HDF and to all of you for tuning in. Thanks for being here. Today's webinar is exciting because you will get to hear from someone that I trained with in the lab. Toward the end of my postdoc, I was interested in testing my hypotheses in fruit flies, and I was incredibly lucky to be able to go to Texas and train with our speaker, Dr. Juan Botas. He works at the Baylor College of Medicine in Houston, and Juan is a pioneering geneticist and has created many of the fruit fly models of neurodegenerative diseases that are used around the world today. Juan is interested in understanding the molecular mechanisms driving pathogenesis in various cell types to identify therapeutic targets. To understand this, he integrates both computational methods with wet lab approaches using a combination of experimental model systems such as flies, mice, as well as cell models. So as I mentioned, he created many fruit fly models of various diseases, including Huntington's, spinocerebellar ataxia, Parkinson's, and Alzheimer's. And all of these models show late onset with progressive neuronal degeneration and disease-specific neuropathology. So he uses these fruit fly models as a discovery tool. So together with state-of-the-art robotic instrumentation, they allow him to carry out high-throughput genome-wide genetic screens to identify genetic modifiers and therapeutic targets. And today he's going to share some of that work with us. So before we start, I want to remind everyone, you can use the Q&A box throughout the talk. So try to get the Q&A box, not the chat box, because I'll be looking at the Q&A box. We should have some time with our speaker at the end for a Q&A. So send your questions in as you think of them. We don't want you to forget, and we'll get to as many as we can. And also as a note, this will be recorded and will be available on our website as well as our YouTube channel in about a week or two. So if you want to watch it again, or if you want to share it with someone who maybe couldn't make it today, it will be available for that. Thank you all again for tuning in, and I will pass it over to Juan.
Thank you, everyone, for joining us for today's webinar. I'm really excited to tell you about our research on Huntington's disease. But first, let me say a few words about myself beyond what Sarah already said, or I would just maybe reiterate that. I'm a geneticist and a neuroscientist working here at the Neurological Research Institute, which is a joint venture between Baylor College of Medicine and Children's Hospital in Houston. But I actually grew up and went to college and did my PhD in Madrid, Spain, under the supervision of a great geneticist, Antonio Garcia Villido, who was a foreign member of the US National Academy of Science, as well as a member of the British Royal Society. During those early years of my training, I had the great opportunity to visit the MRC Laboratory of Molecular Biology in Cambridge, England, and I got acquainted with some of the great British scientists that work at the MRC in Cambridge. After finishing my PhD in Spain, I moved to Stanford, to the Stanford Medical School in California. And again, I was lucky because for my postdoctoral training, I trained in the legendary biochemistry department with David Hogness, Paul Berg, Arthur Comber, and other Nobel Prize winners that were the faculty of that department. And after finishing my postdoctoral training in Stanford, I was recruited to Baylor College of Medicine in the Texas Medical Center, which really was the perfect place and environment for me to move from studies of how embryos develop, studies of developmental biology, to studies to try to understand disorders of the brain. I knew from my training years that the fruit fly has been very successful in teaching us how an embryo forms. And I wanted to study the other end of the life spectrum, to see when things start not going well, when neurodegenerative disorders kick in. I wanted to see if the fruit fly would be useful to highlight what is going on, given its incredible ability, the incredible ability that we have with fruit flies to work with genetics. So what are my research interests? Of course, I'm interested in Huntington's disease, and I'm interested in aging. But not only that, I'm interested in comparing the mechanisms and the similarities between different neurodegenerative conditions. And so, as Sarah mentioned, we not only work on Huntington's, we work on disorders that affect many people, like Alzheimer's, with Parkinson's disease, and we also work with other polyglutamine disorders like Huntington, but that are even rare, like spinocerebellar ataxias. So I'm interested in understanding what are the common themes for many reasons. But one important one is that, of course, if we find common mechanisms between something like Alzheimer's and a rare disease like Huntington, all the incredible resources and effort and money that goes into Alzheimer could also benefit Huntington's disease. So I'm trying to define those mechanisms of pathogenesis and presumably those therapeutic targets that are common to both. Now, our goal, of course, is to develop effective treatments for Huntington's disease. And we share the enthusiasm and the optimism for the approach of using antisense oligonucleotides to reduce the production of mutant huntingtin protein that I'm sure many of you are familiar with and that you have heard in previous webinars. But even if this is completely successful, the history of medicine teaches us that for many disorders, we need a combination of approaches to effectively combat and defeat disease. And some examples of when we use combinatorial approaches, therapeutic approaches, are HIV/AIDS, which typically involve a combination of at least 3 antiretroviral drugs. In many types of cancer, we require a combination of chemotherapies to increase the effectiveness of the treatment and reduce the chance of developing resistance. Hepatitis C is another example in which we use a cocktail of drugs. Rheumatoid arthritis and in general autoimmune diseases. In there, we also use combination of drugs to help manage symptoms and to control disease progression. So as we move towards an effective treatment for Huntington disease, I think it's important to have several bullets. One of them could be the antisense oligonucleotides, and we're all counting on that. But I think we should develop punch number 2 and punch number 3 so that we are able to defeat Huntington's disease. As Sarah said, we work with computers, we work with fruit flies, we work with mice, and we work with humans in neurons. But again, the fruit fly is our discovery tool. And that is because using fruit flies, we can do 1,000 or more experiments with the same time, investing the same time and effort than we would do one experiment in mice. Or we could do 100 experiments in flies with the same effort that we could do an experiment with neurons in culture. So that ability to do high throughput experimentation together with the unparalleled activity to modify the genome of the Drosophila makes it an organism for discovering, for assessing possibilities, for probing therapeutic opportunities. And once you have gained evidence that that is possible, then you move to the other systems. So here is Drosophila in all her splendor, a real picture of Drosophila, a male Drosophila in this case. And again, the importance of this organism is his high throughput experimentation. But Drosophila was first known because of his contributions to our understanding of how an embryo develops into from a single cell after fertilization. So we know that the genes that dictate embryogenesis in fruit flies are the same genes that dictate embryogenesis in the human embryo. As a matter of fact, the genes that tell tell the body what body parts are going to mate where are the same in the flies and in the human embryo. And those genes that dictate where arms or legs are going to form, or wings or legs in the case of the fly, are the so-called Hox genes. And the discovery of those genes are the conserved functions from flies to humans earned the Nobel Prize to one of my American mentors, the late Edward B. Lewis, who gained the Nobel Prize for discovering these genes. Now, fruit flies not only have genes that are conserved between humans and with humans, but also have a brain and nervous system. The brain and the nervous system of the fruit flies has 200,000 neurons, which is still a lot, but of course, many less than the 85 billion neurons that our own brain and nervous system has. But the important thing is that the way those 200,000 neurons in the fruit fly work are the same ways that our own neurons. They connect with one another, they form circuits, and the output of these circuits is behavior. Me moving my hands, speaking to you, you integrating what I'm saying, and rationalizing the arguments that I make. The output of those circuits is behavior in flies and in humans. And the way that the neurons communicate is also very similar through electrical impulse, action potentials, and through the action of neurotransmitters. Now, when I was training, as I said, the fruit fly was known for its contributions of us understanding development. But I had the idea, as I mentioned before, that the same tools, the same advantages of the fruit fly could be used to understand neurodegeneration. And so we created, as Sarah mentioned, a number of models of neurodegenerative conditions in fruit flies. The first one was a model of a spinocerebellar ataxia type 1, and we published this first screen showing that you could suppress neurodegenerative phenotypes, neurodegenerative pathology in the fruit fly, in the attacks in the SCA1 fly, and that those phenotypes could also be then suppressed in the mouse. So the predictions from the suppressors in flies translated to the mouse. But soon after doing that model of the SCA1 fly, I was really attracted to Huntington disease. And in part, that was because of the enormous gravitational pull of Nancy Wexler that got me and many other scientists interested in Huntington's disease. And so we generated a model for Huntington's disease by introducing into the genome of the fruit fly the human gene that triggers the disease in humans. And when you do that, the fruit flies look perfectly fine when they are young. And then around mid-age for flies, which is about 10 days, they develop, or even earlier, they develop symptoms. They have onset. And from then on, there is progression of those symptoms. And at the end, when they are at the end of life, they have this behavior, this uncoordinated behavior, which is somewhat reminiscent of Huntington's chorea. So they have late onset, the disease is progressive, they have choreic-like movements, but also if you look at their brains, you see neuropathology that you also see in the human brain. You see the formation of inclusions, in nuclear inclusions that you can see here in this. Let me get the pointer. You can see the nuclear inclusions containing huntingtin aggregates together with other cellular proteins. So the nuclear aggregates observed in the fruit fly are, as far as we can tell, molecularly identical as the nuclear aggregates that we see in the patients, in the brains of patients. So they have also a similar neuropathology. Once that we had this model of Huntington's disease, we published a slew of papers demonstrating that we could use the sophisticated genetics of the fruit fly to actually ameliorate disease in the fruit fly, to suppress the pathology of Huntington's disease, and to study the mechanism by which mutant huntingtin creates havoc in the neurons of the fruit fly nervous system. And we were able also to distinguish pathogenic and compensatory transcriptional changes that occur as a consequence of Huntington's disease, so that we can use those compensatory changes as also potential therapeutic targets. So the point that I want to make is that we use these models in a number of studies to show that in principle, the pathogenesis, the pathology of Huntington's disease, at least in flies, it could be ameliorated. And actually, we were able to show that, as I mentioned for the case of SCA1, that some of those genetic manipulations that ameliorated pathogenesis in the fly could also ameliorate pathogenesis in the mouse. But where else can we find genes that confer neuroprotection? One good place to look is the genome of supercentenarians. Supercentenarians are those people that get to live over 110 years old. Semi-supercentenarians are those people that get to live over 105 years old. And what you're seeing here is the picture of this woman, Jeanne Calment, a French woman, who, according to records, is the longest living person ever. She lived up to be 122 years old. And you see her here on the right picture, celebrating her 120th birthday. And on the picture on the left is the same woman a century before, 100 years before. So what is special about these people like Jen, or actually, what is special about Jen that allowed her to live so long and actually be sharp, mentally sharp, until very, very advanced ages? This person lived around 50% more than most of us would live, or maybe 60% more. And that is, as impressive as it might be, it is something that nature has learned to do very often. For example, we have here an image on the left of a common rat whose lifespan is typically 2 years, but another rodent, a cousin of the common rat, which is the African naked mole rat, can live more than 30 years. So nature can tweak lifespan and multiply that by at least 10 times in related species. And that means, obviously, not only that this ugly naked mole rat can live 10 times more than the common rat, but actually that their brain stays sharp for all those many years. Even more impressive than that, we know that there are ants with incredible longevity between casts of the same species. So in some species of ants, males live for days, worker ants live for months, and queen ants can live for up to 30 years. So within the same species, nature is able to tweak lifespan and neuroprotection to multiply lifespan and reproduction by more than 1,000 times. But let's go back to the human case. Aging, of course, is the #1 risk factor for the onset of all the neurodegenerative conditions. However, not everyone who reaches old age develops dementia. In fact, those supercentenarians, these exceptional people, demonstrate the ability to avoid or delay neurodegeneration. What you're seeing now on the left is a picture of the oldest person that currently lives. She's 117 years old now. She's celebrating her 115 years old in this picture, and she lives in Spain. And if you read the New York Times, you might have seen a couple of days ago the news about Maury Markov, which was an American supercentenarian that passed away only a couple of days ago. And he was also, like Maria, extremely sharp mentally until the very last days of his life. He donated his brain to science, like other people have done in the past. And what is amazing is that some supercentenarian people that remain sharp until the very last days of their life when we examine their brains, we can see signs of pathology, signs that are typically associated with Alzheimer, like Alzheimer plaques. But despite that, their brain remain cognitively intact. So these are examples. These centenarians or super-centenarian peoples are examples of the ability that has the resilience ability that has the brain of at least certain individuals to resist dementia and to resist neurodegeneration. So why are certain individuals protected from the dementia or from the aging affecting the brain? The answer is that the brain is naturally resilient against disease, aging, and injury, and tries to combat against these offenses. Similar mechanisms of resilience are also present in Huntington's disease, in which you can see that people that having the same mutation can have disease onset separating them by 20 years or more. So we know that there is also resilience at play in the case of Huntington's disease. So, of course, there are environmental factors and lifestyle factors at play, but understanding the genetic factors of resilience demonstrated by supercentenarians are demonstrated by the difference in onset in Huntington disease could lead to better therapeutics to protect against conditions such as Huntington's. So to investigate the genetic basis of supercentenarian resilience, we obtained data from an Italian supercentenarian and semi-supercentenarian cohort. There are less than 300 living supercentenarians in the world, and this cohort provided data for approximately 20% of them. Now, what is the problem with supercentenarian or semi-supercentenarian data? The problem is that it's so scarce. There are so few individuals that reach that super advanced age. And so the data that we can obtain from them is very limited. And so, because all of us, every one of us in our DNA have millions of variants. The supercentenarian people have millions of DNA variants. And looking for those specific variants that actually are driving that extraordinary longevity and neuroprotection is literally like looking for a needle in a haystack. So given that that data is so rare, the sample size is too small to extract information using traditional methods, association methods, like genome-wide association studies. Therefore, we used a series of integrative approaches to improve the detection of genes related to longevity. We use traditional in silico analysis like genome-wide association studies, transcriptome-wide association studies, and we also work with my collaborator here at Baylor, Dr. Lichtar, to obtain evolutionary action scores, which can improve the detection of causal variants using small samples. But most importantly, we use data from lifespan extension studies in animal models to further implicate the role of these DNA variants in longevity. So essentially, we work to layer evidence that a gene was related to longevity piece by piece. For example, we could take a low confidence but trending GWAS variant and assess if the DNA variant was related to gene expression changes with TWAS or alters protein function based on evolutionary action. And then we could assess whether a concordant manipulation in an animal model resulted in increased lifespan. In this way, we could gain evidence to implicate genes related to lifespan extension in supercentenarians that otherwise would not be possible. To facilitate discovering supercentenarian genes, now we built a cross-species network of genes implicated in lifespan extension in animal models, including yeast, including worms, including mouse, and including flies. And we use this network of lifespan extension in animals to filter the low-confidence supercentenarian candidate genes. And then we layer these results with EA. Briefly, evolutionary action. Evolutionary action is an algorithm, and I'm going to try to explain it with simple words here, developed my collaborator, Dr. Lichtar, that weighs the impact a DNA variant has on gene function by assessing how often a specific amino acid substitution happens at a conserved residue during evolution. So substitutions that are rare have high EA evolutionary action scores, and we would be predicted to have a larger impact on the function of the protein. We found that a considerable number of supercentenarian GWAS coding variants have a high EA score. And many of these, actually, with high scores, also map to gene related to lifespan extension in the animal model network. So, by integrating a variety of computational approaches, GWAS, TWAS, evolutionary action, and machine learning, we were able to identify a significant number of gene candidates implicated in the extreme longevity phenotype of supercentenarians. And to further explore the mechanism of resilience against neurodegeneration, we assessed these genes for their impact on HD pathology using the fruit fly model system. Why fruit flies? Again, because they allow for the high throughput experimentation, so we can test many possibilities. And so it's not just the intrinsic ability of the fruit fly to do high throughput experimentation, but actually we combine them with robots, we can even increase further the throughput. So what you are seeing here is a robot handling the flies, and the robot has a camera that films the fruit flies as they move, and the computer within the robot actually tracks the trajectories of each individual fly, as you saw in this video. So that if you compare healthy flies with Huntington disease flies, after onset, the Huntington disease flies start moving with a slower pace, more sluggishness. And so then we extract the movement metrics from this exercise so that we can plot motor performance of healthy flies here in black, or Huntington disease flies as a function of time as they age. And as you see, the healthy flies move very quickly, very well, and until late in life, as you see by the bar, by the black graph. But Huntington disease flies move well when they are young, before day 6 or 7, and then they start performing poorly in these behavioral tasks. And this is due because impaired neuronal function. So using this screen, using these fruit flies and this behavioral assay, we were able to examine or to screen all those supercentenarian candidates that were obtained from the computational analysis, and that were also experimental modulators of lifespan in animal model systems. And we discovered the interesting, the super exciting thing is that we discover many of these genes identified by supercentenarian variants that were, when tested in the Huntington disease model, were able actually to suppress the pathology of Huntington disease in neurons. They were able to suppress the pathology of Huntington disease in glia. And in some cases, they were able to suppress the pathology of Huntington disease in neurons and glia. And I want to show you a specific example of one of these suppressors of HD Huntington disease-related pathology that originates from a discovery of a DNA variant in the supercentenarian people. One of these DNA variants is in a gene called ENSRR, which is an insulin-like receptor in humans. And when we modulate the activity of the corresponding gene in the fruit fly, we see that, in contrast to the behavior of Huntington disease flies that you see here in blue, that move well when they are young, but then they decline as they age. These Huntington disease flies with this intervention, having an intervention in these supercentenarian genes, now they move much more in a much more agile way, higher speed, less sluggish, less turns, less stumbles, resembling more how a Huntington disease flies moves. So genes implicated in lifespan expansion and neuroprotection in supercentenarians can mitigate HD-related pathology. So in conclusion, we have identified genes associated with extended lifespan and resilience in supercentenarian and semi-supercentenarian population and animal models. And genes associated with lifespan extension are significantly enriched for suppressors of Huntington disease and actually also for other neurodegenerative conditions that I have not shown you. And I want to finish my presentation and allow you to ask me words. But first, I want to acknowledge my team, my collaborators. Science is always a team effort. And we could not have done this without not just my lab members, particularly Megan Mayer, who actually was the person that drive most of the project that I have told you, but also with the help of my collaborators, Dr. Lichtark, that developed these computational tools that are able to extract value from a small sample size. And of course, our Italian collaborators that allow us to work on these Italian supercentenarian people. And I'm gonna finish here and give it back to you, and I'm very happy to answer any questions you might have.
Thanks so much, Juan. That was fabulous. As you know, I think fly work is super fascinating. We can learn so much from them, these unexpected little creatures. So we have questions that are popping up in the Q&A. Please keep adding your questions to the Q&A box during the Q&A session. So I think I'm just going to start at the top and we'll go down. So somebody asked, would doing puzzles and solving math problems keep the brain healthy by keeping it busy, and would that help to slow dementia?
That is a very good question. And the answer is yes, because there is correlation between people that challenge their brains and the incidence of dementia. But the but here is that it has to be a real challenge. So probably doing a puzzle or something that your brain can do easily is not enough. You really need to challenge your brain. Examples of how can you do that? You could do, for example, learning a new language. That is a real challenge. You really need to engage your brain and push it. You can learn how to dance if you don't know how to dance, right? You need to integrate music with your body movement. It has that works, but you really have to push it for it to work. Otherwise, it doesn't.
I love that. Thanks, Juan. So here's another question. What are the genes that modify longevity in ants? Why not use these animals since they are presumably genetically more similar to each other than humans?
So we have a bird's eye view of the genes that are able to modify lifespan in ants. We know, for example, that the workers in ants that are workers, their genes involved in metabolism are more active than in queen ants. On the other hand, in queen ants that live so much longer and whose brain remains intact much longer, we know that our genes involved in cell preservation, in recycling of cellular debris that are more active. So we know those generalities, but we don't know actually what are the triggers that actually orchestrate that different gene activity. And that's what we're trying to find out.
So interesting. Yeah, that was, I think, my most interesting fact learned today, that any ant could live for 30 years. So thank you, Juan. There's a question from Jenny Morton. She asked, do the modifiers that ameliorate HD phenotypes extend the lifespan of normal fruit flies?
That is also a very good question. We are testing that possibility. So the vast majority do not. The vast majority do not. But we have some preliminary data suggesting that in some very exceptional rare cases, we can manipulate genes to extend the lifespan of healthy non-disease flies. Now, those are the rare exceptions. The norm is not that.
Interesting. So here's a question. Are the genes you have investigated published somewhere or otherwise available to the scientific community?
The genes that we have published mitigating Huntington's disease in flies, the majority of them are published. And I showed some snippets in my presentation of some of those papers. The genes that come from our analysis of supercentenarians, they are not published. We are actually putting this publication together as we speak. So what you have heard today is all of supercentenarians, is all unpublished. But
That's great. I love.
But it will be published very soon, I promise.
Exciting. So someone asked basically about diet. They asked, since so many of the supercentenarians are from Italy, is there something about the Mediterranean diet that's neuroprotective?
Well, yeah, I mean, I think there is a lot of clinical evidence about the importance of diet, right? And particularly of the Mediterranean diet. I think we listen to those clinical associations all the time. So there is some truth in there. Lifestyle is important. Diet is, and diet is, of course, is part of the environment and lifestyle. But genetics is also important. And so of neuroprotection by looking at these people, obviously, but everything contributes. Everything is an interplay between genetics and environment and lifestyle and diet and exercise and all of that, of course.
Yeah, I think that's a good plug, especially for people living with HD, that living a healthy lifestyle through diet and exercise and all of these things is really the only thing that we have evidence that can really help keep you healthier longer. So eating a healthy diet and exercise really goes a long way. Here's a question. They asked if the gene FAN1 was seen in supercentenarians.
Not something that we have picked up.
Interesting. Okay. So this person says, brilliant. Where in the flies was the INSLR? I'm thinking they mean the insulin receptor gene. Where was it reduced? And do you envision any behavioral modification which could affect HD?
Manipulated the activity of the corresponding fly gene in the whole nervous system. And by doing that, then we ameliorated that motor performance, that movement impairments that we define in the fruit fly, including what I showed in that graph, the speed at which these flies move. But not only that, I didn't show data for stumbles, for turns that could be a measure of disorientation. All of those movement metrics are improved by reducing the activity of the gene in the neurons of the fruit fly. And I should say, both in fruit flies that express an N-terminal fragment of huntingtin, as well as in fruit flies that express a full-length huntingtin human protein.
Thanks, Juan. So here's a question they start. Great talk. You mentioned that Super-Ager brains have plaques and tau, but no dementia. Based on that, do you think plaques and tau are the result and not the cause of aging diseases?
No, I didn't. I don't want to imply that. I wanted to emphasize that the brain is naturally resilient, and some brains have more resilience capacity than others. And I showed those examples. I mean, it could be that there are supercentenarian brains that don't have plaques, but I wanted to show an example of a supercentenarian brain that had plaques, and nevertheless, it was the resilience of that brain that allowed that that individual to be sharp despite having plaques. So I mentioned that because the goal, or my goal here, was to identify those factors, those genetic factors that confer those resilience, that allow to combat the plaques, right? That allow to combat neurodegeneration, that hopefully will allow to combat Huntington's disease as well.
Thanks for clarifying one. There's another question actually about those, the plaques. They ask, are the plaques in the supercentenarians the same size and frequency of HD or Alzheimer's brains?
I really don't know, and I don't think it's well known, simply because there are probably only a handful of supercentenarian brains with plaques. And so from what I know, they look very, very similar, but again, I couldn't tell you whether they are exactly the same or not. There are too few of them.
Thanks, Juan. So here's another question from Jenny Moore, and she says, Juan, I should have said that I enjoyed your talk very much. Another question, is there any overlap between the longevity genes and the HD modifiers you've already published? If so, could you tell us more?
Yes, there is an overlap. there is an overlap. And actually, when we started analyzing the supercentenarian genes using this combination of in silico approaches, we developed a list of candidates, and we wanted to screen them on the Huntington disease fruit flies. But the first thing we did was to test, okay, what have we screened already? And that's when we found that indeed some of those that came from the supercentenarian analysis, we had a screen in the context of other projects in the Huntington fly. So there was an overlap. And that's what I want to actually dig in deeper and assess what is the real overlap, how many of them are there, not just with Huntington's, but also with Alzheimer's.
Maybe I'll take prerogative and add to that. Have you tested any of those in mice yet, the ones that overlap?
We have not tested them in Huntington's disease mice. We have tested some in a human neuronal precursor cell model of Alzheimer's.
Did they change the phenotype or?
Yes.
Exciting. Cool. Very cool. Okay, so here's another question. Thanks for an excellent talk. What is your view on insulin signaling by gut microbiota?
Wow. We are learning. I don't have a particular view of that. I am impressed and at awe, like everybody else is, about the influence of microbiota on the brain, because it's something that we essentially had no idea just a few days ago. But that actually goes back to what we were speaking before, which is the influence of the environment. I mean, the microbiota is in part of our environment, right? And so it's another example of how we need to take care of ourselves if we want to keep healthy. Yeah.
Thanks, Juan. So here's an interesting question. Have you analyzed families of these supercentenarians? I'm assuming they mean for genetic similarities.
No, I don't have data from families. I would like to have data from families. I would like to have data from Huntington disease families also, so that we could actually use the same computational tools to assess what factors confer resilience before what we know already that relates to somatic expansion. But we are using some of the available data that is becoming more and more available through the British Biobank or through the All of Us project here in the US, that includes people that reach advanced ages, not very few supercentenarians there, but we can obtain data from people that are in their 90s or high 80s, and maybe some of that could be useful as well.
Excellent. So I just want to let people know, keep asking your questions in the Q&A box. We'll get to as many as we can. So here's someone who's curious about your fly models. They ask, was there any difference in the CEG repeat length within the flies, or what was the CEG repeat length that you used?
So we have made flies with many CEG repeat lengths. As you might imagine, as you might predict, the severity of the phenotype, the onset of the disease, the speed of the progression depends on the repeat length. Well, actually depends on repeat length also, and on one, and second, it also depends on the level of expression of that protein that you produce with that repeat length. So the more repeat length, the more severe, the earlier onset, the higher level of expression, also the earlier onset and the more severe phenotype. So at the end, we chose a repeat length, which is longer than you would see in humans, because that would also facilitate our high throughput screening, because we wanted to have flies that had no onset early on when they were young, but they would get the onset relatively early, so that then we could do experimentation at higher throughput. So we chose repeat lengths that go between 60 and 80, or in some cases we went even farther up.
Yeah. So I think this is a clarification question, and I think there's a typo that's preventing me from fully understanding it, but they ask, thanks for the presentation. If I understood, the supercentenarian genes are interfered in Huntington's disease. So perhaps she means that the supercentenarian genes can interfere with Huntington's disease in the flies. So maybe you could kind of explain that a bit about the genes that you tested.
Yes, so interfere, I wouldn't use the word interfere, because it sounds like it's making things worse. And what I'm trying to find out here is whether we can identify supercentenarian genes that confer this extraordinary individual's resilience and neuroprotection. I want to find those factors and then test them in Huntington's disease. In that sense, they interfere, but in a positive way. So the goal here is to have to find factors from supercentenarian genetic variants that are able to be protective in the case of a disease like Huntington's, and at least in flies that appears to be working.
Cool. So here's a question that asks about the brains that you used from the supercentenarians. They say, with such a small number of supercentenarians who are currently living, how many brains of those who have died are being looked at? It's such a special tissue.
Yes. Well, we are not looking at the brains. We are looking at the genome of these people, right? But I saw some examples of supercentenarian brains that. Of people that have donated their brains, like this, like Morris, this supercentenarian American male. By the way, supercentenarian males are much more rare even than females. Most of the supercentenarians are women, of course. But we have their genomes, and that's what we are studying, their genomes. Right? And in some cases, we would like to complement that by studies that look at how gene expression changes with age, and for which we have data also, not from supercentenarian people, but from people when they're young, from people that when they're middle-aged, from people that we are old. And we have the corresponding data from flies. We have gene activity at the level of the transcriptome from young flies, middle-aged flies, old flies, proteomic data from young flies, old flies, and proteomic data from young HD flies, old flies. So integrating all of that with the supercentenarian data, I'm hoping that will give us further insight.
So do you see a lot of changes as flies or people age, their gene expression profiles change quite a bit?
Yes, they do. They do. There are genes that go up with age. There are genes that go down with age. Right? There are genes that go up in disease. There are genes that in disease, they seem to have a signature of accelerated aging. And there are genes that appear to have compensatory activity, trying to fight back some of these aging mechanisms. So yeah, I think it's very interesting. And complementing that with genomic data, I think could be very fruitful.
No pun intended, right? Very interesting.
You
So,
got it.
so here's a question. They essentially say, I take it there were no obvious smoking gun gene variants in the supercentenarian. So maybe was there one gene or a few genes that you found above all others that had a very large effect or the largest effect?
Well, I mean, as I mentioned, uh, what I presented is all unpublished. We are still digging. We are still validating results. Probably my guess is that the effect, the neuroprotective effect of these supercentenarian variants is additive, or there are several, perhaps many variants that contribute to resilience, and some of them might have a significant effect on their own, and some others might work together. But we are testing them at this timing point individually in flies, and as I showed you with the insulin-like receptor, that on its own is able to confer neuroprotection. But with fruit flies, actually, we are planning also to combine variants and test not just one, but more than one in combination. That oligogenicity that I think is difficult to model, but I think the fruit flies are particularly well adapted to do that. And so that's part of our future plans.
Very cool. Have you, by any chance, looked at the changes in gene expression and compared that to, oh, actually, you did. You compared it to the GWAS from the human data sets, right? Yeah.
Yes. We are trying to compare, are there variants in GWAS that might be even not significant, but suggestive that are in genes that we see alterations in transcription or alterations in proteomics as a function of age. All that integrative work, I think, could be also useful to identify factors that change with age, some of which might confer resilience.
Oh, cool. So here's a question from Christopher Pearson. Wonderful talk. Thank you. How much somatic repeat instability arises in the flies, if any repeats, for any repeat, CAG, CGG, or any other repeat?
I am not sure, Chris. I mean, we have obviously investigated that because we were obviously impressed by the work that you and others have done on somatic instability. As far as I can tell, based on what we have done, the somatic instability in the fruit fly is much less than the somatic in the fruit fly brain, at least, is much less than what we can see in the human brain. And so from that data, I suspect that the modifiers that we might find in the fly might correspond to mechanisms of pathogenesis that for the most part do not depend on somatic instability, but other factors that contribute to pathogenesis.
That's interesting. And I think in relation to that point, right, like you were pulling out genes from the supercentenarians and cross-referencing those to your flies. And if they don't have high somatic instability, that might be why you're not pulling out all of these DNA damage response genes, all of these very top modifiers that we see in human studies. And so you might be going after mechanisms that are unrelated to somatic instability and not necessarily.
It could be, but bear in mind that before doing experiments in flies, and I think that was really the key of what we did, was to integrate the in silico analysis of supercentenarian data with all the network of genes that have been discovered by all researchers working with yeast, working with worms, working with flies, working with mouse. We created a network of life-extending genes, cross-species, and we compare the supercentenarian data with that cross-species life-extending network before testing them further in flies. And so I think that in that network of longevity extension from cross-species analysis, there are few, if any, genes implicated in somatic extension. But I could be wrong about this. I need to check that back.
That's exciting. That's super cool. So here's a question from Megan Donaldson. She says, since you've been able to show that the HD flies, which have been modified with supercentenarian genes, and they perform better than the HD controls, how much more work do you need to do on the flies before you can move on to larger animal models or human iPSCs, stem cells? Really, how much do you eventually, or would you see this eventually moving to being therapeutic for humans?
Well, the first thing we're going to do is to move it to iPSCs, Huntington disease iPSCs, and so I think that would be the first move forward. But I want to continue using the fruit fly as the discovery tool, as the exploratory tool, because I think the confidence on a target comes not just from validation outside the species, in this case flies, or moving from flies to mouse or to human neurons in culture, but actually comes from analysis of the mechanism and pathways that you discover. So we find resilience genes that all map to a specific molecular mechanism, to a specific molecular pathway. Then we will be much more confident about that particular avenue, therapeutic avenue, that if we have one isolated target that has no biological connections with other functions. And so exploring in breadth mechanisms, pathways is important, but then moving and jumping from the fly and to the mouse or to the human neurons in culture, which is the first thing we're going to do, is obviously very important.
Awesome. Thanks, Juan. I'm really looking forward to reading this paper. This is going to be very fun to read. So it looks like we're at 1 o'clock. We're at the end of our time, so I'll wrap it up there. If you had a question that we didn't get to, I'll send those to Juan, and if he's willing, he can follow up with you directly. But I want to end by, again, thanking our sponsor for this webinar, Neuron Biosciences, as well as everyone who donates to the HDF to support the research that we're able to fund. And of course, our audience, and last but certainly not least, Juan, thank you so much for joining us today.
My pleasure. Thank you, all of you.
Our next webinar will be Thursday, September 26th. We're going to have a bit of a break for the summer, and then we will be back to recap HDF's biennial conference that's happening in August. There's going to be lots of exciting research to cover in September. Thank you all so much for joining us, and I hope to see you in September.